Let's understand the hardest topic for everyone, especially for medical students. This topic named Krebs cycle, as also known as tricyclocarbocyclic acid cycle or Krebs cycle, biochemical pathway used to generate energy through oxidation reactions, and in this process take part in acetyl-CoA, synthesis of NADH, production of amino acids. The cycle takes part of the eukaryotes in mitochondria, organella, and the cytosol of the prokaryotes. The first step involved is actually preparation step, is for glucose, which has 6 carbon atoms. Pyruvate is derived through the glucosis process from the glucose, which has 3 carbon atoms. The next step includes the oxidation from pyruvate to acetyl-CoA by the enzyme pyruvate dehydrogenase. And in this reaction is generated molecule carbon dioxide and molecule NADH with the 3 atoms of carbon in a pyruvate molecule. The next step is acetyl-CoA is combined with the molecule oxaloacetate, which has 4 carbon atoms. The resulting of adding oxaloacetate molecule to acetyl, which has 2 carbon atoms, it gives 6 carbon atoms, which the molecule named citrate. This reaction is catalyzed by the enzyme citrate synthase. The next step is the molecule of isocitrate is oxidated into alpha ketaglutarate by the enzyme of isocitrate dehydrogenase. And in this reaction, a molecule of NAD is reduced to NADH molecule. And also in this reaction is generated the molecule of carbon dioxide. Hence, the alpha ketaglutarate molecule has 5 carbon atoms, is lost because of the generating carbon dioxide molecule in this reaction. The next step is the molecule of alpha ketaglutarate is converted to succinyl-CoA by the enzyme alpha ketaglutarate dehydrogenase. And in this reaction is also reduced NAD molecule into NADH molecule. In the samely, this reaction acetate to alpha ketaglutarate released carbon dioxide molecule. The molecule of succinyl-CoA has 4 carbon atoms compounding the molecule. The next step is the molecule of succinate molecule is generated by succinyl-CoA by the enzyme of succinyl CoA. And in this reaction is generated the molecule of GTP molecule. The next step is the molecule of succinate is converted into the molecule of fumarate by the enzyme of succinate dehydrogenase. And this molecule has 4 carbon atoms, and in this reaction is generated the molecule of QH2 molecule, which is used for the production of the molecule of FADH2 molecule. The other step is the molecule of fumarate is converted into the molecule of malate, which this molecule has 4 carbon atoms, and this reaction is occurred by the enzyme of fumarase. Eventually, in the last step is the molecule of malate, which has 4 carbon atoms, is converted into the molecule of oxaloacetate, which has also 4 carbon atoms. And this reaction is occurred by the enzyme of malate dehydrogenase. And in this reaction is also, like others, is generated the molecule of NAD, molecule to NADH molecule. Now let's look the resulting of the whole Krebs cycle. Here is the generated molecule is 1 molecule of NADH2 molecule and 1 molecule of FADH2 molecule, and also 1 molecule of QH2 molecule. And in these reactions is generated also 2 molecules of carbon dioxide. All the molecule of NADH2 and FADH2 molecules are generated for giving the energy, giving the electrons transfer, which is generated the molecule of ATP molecule in the mitochondria in eukaryotic organisms. So it was the short introduction or understanding of the whole Krebs cycle. This is the end of our video. Thanks for attention.